Hello and welcome. In this video, I'll introduce DAQ-Score, a powerful computational tool that uses deep learning to evaluate the residue level accuracy in protein structure models from cryo-EM maps up to 5 angstroms in resolution. Using this score, researchers can easily identify areas in the structure model that need correction, allowing for more accurate structure models. The DAQ-Score pipeline starts with a deep learning model, which scans the cryo-EM map to predict the probability of amino acids, secondary structures, and C-alpha atoms at each position. Then, by comparing these probabilities with the protein structure provided, we compute log-odd score for each residue in the model. So the way that this works is first, we compute the probability for the amino acid type at residue I using deep learning. Next, we normalize this probability by computing the average probability of this amino acid type across all atom positions in the protein model. We take the log of this ratio, giving us the DAQ score. Finally, these computed scores are averaged with a window size of 19. In general, high DAQ scores indicate good agreement and lower scores indicate errors. We constructed a database of cryo-EM derived structures on PDB that were revised and have two versions. The residues that were changed between the first and second versions were assumed to be incorrectly modeled, and the residues that remained unchanged were assumed to be correctly modeled. We then computed the DAQ score and visualized the distributions in these two classes. Here, red is the distribution of DAQ scores of incorrectly modeled residues, and blue is the distribution of DAQ scores of correctly modeled residues. You can clearly see that below DAQ score of negative 0.5, all of the residues are incorrect. This means that if you see DAQ score below negative 0.5 in your protein model, then there is most likely a modeling error. To get started, visit em.kiharalab.org and scroll down to the roadmap section. Here, select DAQ score. Now you'll need to prepare your input files. You'll need a protein structure file in .pdb format and a cryo-EM map in .mrc format. Ensure that your map resolution is within the supported range of up to 5 angstroms and that your protein is aligned to the map. On the DAQ score page, upload your .pdb and .mrc files. Then hit the upload button to upload and submit the job. This is the page where you will see the results once the job is complete. If you're logged in, you'll receive an email notification when the job is done. You can then click Job Manager, My Jobs to view your job results. Otherwise, bookmark this page or write it down somewhere and check back later. After the job is processed, return back to this submission page. Scroll down and you'll see a 3D model where each residue is colored by its DAQ amino acid score. Blue regions indicate high scores, meaning that these areas fit the map well. Red regions indicate low scores, flagging potential errors. By clicking this button, you can download the structure in .pdb format and view it in tools like PyMol. The DAQ score is written to the B factor column in the PDB file, so in PyMol you can run this command to get the same color scheme. In the website, you can also click Show Map to superimpose the cryo-EM map on top of the structure to visually check the map model agreement. We've also pre-computed the DAQ score for every single protein model on PDB that's derived from cryo-EM. You can access this database at daqdb.kiharalab.org. Link will also be in the description. Thank you for watching. To learn more, visit our tutorial page or check out the detailed documentation and resources linked below. To learn more about the tool, read our paper published in Nature Methods. Thank you.